So, pharmacodynamics is the science of how drugs interact with the body and how they actually induce their effect. So, we've all known these sequence. You get sick, you go to the hospital or to a clinic, the doctor gives you a prescription, you go to the pharmacy, and the pharmacy gives you this pill that you take, and eventually you get better. But how do you actually get better? How do you go from being sick, taking a medication like a Pac-Man, to actually getting better? And this is the topic of pharmacodynamics. So, some drugs interact by or activate or induce their effect because of their physical or chemical properties, such as osmotic diuretics that absorb water into kidney tubules, thus increasing urine output. And other drugs interact because of their chemical properties, such as antacids, which go to the stomach and interact or react with stomach acids in order to reduce the stomach acidity. But most drugs interact in a different manner. They interact by interacting with receptors. And receptors are proteins that are usually found on the surface of cells. And when these proteins are stimulated by their ligand, which is the molecule that activates this specific receptor, it causes the receptor to induce a cascade of events leading to, ultimately, the desired effect. So, what are the types of these receptors? So, what are the different types of receptors? And in fact, there are plenty of receptors and there are plenty of ways drugs can interact with the body. But there are four main receptor classes, and these types of receptors are intracellular receptors, enzyme-linked receptors, ion channels, and G-protein coupled receptors. Let's go into more details. So first of all, let's start with the simplest type of receptors, which is the ion channels. So first of all, ion channels. And these are channels that when stimulated by a ligand, they are ligand-gated ion channels, and when they are stimulated by a ligand, it causes the channel to undergo a conformational change, thus leading or causing the channel to open, thus allowing ions to flow across that channel and into the cell along their concentration gradient. And these are ligand-gated ion channels because they are gated, but when stimulated by a ligand, they allow ions into the cell. There is a second type of ion channels that is voltage-gated. And these types of receptors open upon depolarization of the membrane. So when the potential difference between both sides of the membrane approaches zero, goes from a negatively charged one to closer to zero, it causes these channels to open. The third type of receptor, and the slightly more complicated kind of receptor we're going to speak of, are enzyme-linked receptors. And these receptors are composed of a receptor that extends across the cell membrane and has two sides, a cytosol-bound site, or a site that is located in the cytosol or the cytoplasm of the cell, and also has a recognition site. And the recognition site is where the ligand binds. And the recognition site is present on the outside of the cell or on the cell surface. And when a ligand binds to this receptor, it causes the receptor to be activated. And it causes a conformational change that causes both enzymes to be linked together. And when this receptor is activated, it either leads to the activation or deactivation of the enzyme that is present on the inside of the cell. Usually, or at least in most cases, it leads to the activation of a protein kinase, which causes the phosphorylation of this specific protein that is phosphorylated by this specific protein kinase. And the third type of receptor we're going to speak of are intracellular receptors. Intracellular receptors are a bit different from the other types of receptors in that they are fully located on the inside of the cell or on the cytoplasm of the cell. And they require a very lipophilic or a very lipid-soluble ligand, and they require a very lipophilic ligand to diffuse into the cytoplasm and to bind to this receptor. And when a ligand binds to this receptor, it activates the receptor. And this activation causes a conformational change to the receptor or to the protein of the receptor, thus causing it to be activated. And when activated, this protein then migrates, this receptor then migrates to the nucleus, where it binds to the hormone response element on DNA, the hormone response element, or HRE, on DNA. And when it binds to the hormone response element, it causes DNA to form a messenger RNA, which if you've been paying attention in your biology class, you would know that this would lead or would be translated into a protein, and this protein will ultimately lead to the desired effect or to the effect of this intracellular receptor. And the effect of these receptors usually lasts from several hours to up to several weeks at a time. The last type of receptor we're going to speak of are G protein coupled receptors. And these are very, very important because more than 50% of drugs interact or induce their effect by interacting with these types of receptors. And these types of receptors are composed of a receptor or a protein of a receptor that has 7 regions that span across the cell membrane. And on the outside of the cell or on the cell surface, it has a recognition site, which is where the ligand binds. And if you were to take a top view of this receptor, it would look more like if it were forming a circle. Okay, so let's get back to the normal view of this receptor. On the cytosol part or on the cytoplasm, it is linked to a G protein or a GDP bound protein. And this G protein is composed of 3 units: an alpha, a beta subunit, and a gamma subunit. And the alpha subunit is bound to a GDP molecule. And when a ligand stimulates this receptor or activates this receptor, it causes a conformational change that travels all the way into the alpha subunit and causes the GDP molecule to leave its place and for a GTP molecule to take its place. So it causes a GTP to bind in place of the GDP molecule. This in turn activates the alpha subunit and causes the alpha subunit with its GTP molecule to detach from the receptor and bind to the effector. This effector may be an ion channel that's present on the cell membrane, or maybe another enzyme, thus triggering a cascade of second messengers. Now we've said an interesting word, the second messengers. So what are these and what are the different types of second messengers? So let's start with second messengers. And let's start with the 2 similar second messengers, which are adenylyl cyclase and guanylyl cyclase. And adenylyl cyclase converts ATP into cyclic AMP. And this cyclic AMP then activates protein kinase A, which leads to the phosphorylation of proteins. Guanylyl cyclase is similar, except whenever you see an A, replace it with a G. So it replaces or it converts GTP into cyclic GMP. And this increase in cyclic GMP activates protein kinase G, which ultimately leads to the phosphorylation of proteins, because this is a protein kinase which phosphorylates proteins. Third of all, we have phospholipase C, PLC. And phospholipase C leads to the formation of IP3 or inositol triphosphate and diacyclic glycerol or DAG. DAG then activates the calcium-dependent protein kinase, which is also known as protein kinase C. And as its name tells you, it does phosphorylate proteins. IP3 increases or binds to the IP3-gated calcium channels on the endoplasmic reticulum. And when these channels are activated, it leads to the release of calcium into the cytosol, or into the cytoplasm of the cell. And this calcium may either go on to cause a contraction all on its own, cause contraction, or may activate the calcium calmodulin protein kinase. So let's write that down here. And since this is a protein kinase, it would lead to protein phosphorylation. When is adenylyl cyclase activated and when is phospholipase C activated? So what determines the type of second messengers that is employed in the G protein coupled receptors? And this is determined by the type or the class of G protein coupled receptor that exists. So there are three main classes of G protein coupled receptors, or three main classes of G proteins. First of all, there is GI coupled receptors, and the opposite of this class of proteins is the GS coupled G protein. And both of these act on adenylyl cyclase. So if you can see this drawing in the same color as I do, GI inhibits adenylyl cyclase. And if you remember from the previous slide, you should recall by now that less adenylyl cyclase would lead to less cyclic AMP and lead to less protein kinase A, thus leading to less phosphorylation. Whilst the opposite, or GS coupled G proteins, or GS coupled G protein coupled receptors, would activate adenylyl cyclase, and then activated adenylyl cyclase would cause an increased cyclic AMP and would lead to more activated protein kinase A, and would thus lead to more protein phosphorylation. GQ coupled is a bit different because it activates phospholipase C, and if you recall from phospholipase C, it leads to the formation of two molecules, not just one. It leads to the formation of DAG, or diacyclic glycerol, and inositol triphosphate, or IP3, may then activate protein kinase C, or may increase, IP3 increases the calcium levels inside the cell, which may either lead to contraction all on its own, or may cause the activation of calcium calmodulin protein kinase. One interesting feature of this receptor, G protein coupled receptors, is its capability of amplifying whatever signals that come through it. So let's say, for instance, you have just one G protein and you stimulate this one protein with a single ligand. So you have one activated receptor. This one activated receptor may then activate 100 molecules of adenylyl cyclase, and each one of these activated molecules of adenylyl cyclase would cause the formation of 100 molecules of cyclic AMP, and this would activate in turn, each one of these molecules would activate in turn 100 molecules of protein kinase A. And each molecule of protein kinase A can phosphorylate 100 protein molecules. So if you do the math real quick, you could do 100 times 100 times 100 times 100. You would see that you started with only a single molecule, and now you are ending with a figure that has 8 zeros. So that's 1, 2, 3, 4, 5, 6, 7, and 8. You started with a single ligand, and now after amplification and signal transduction from one molecule to the next, you end up with 100 million molecules. 100 million molecules of phosphorylated proteins. This is it for this video. If you like it, thumbs up. If you don't, thumbs down. Please leave your feedback in the video comments, and please subscribe to see more. See you in the next one.